My name is Vicky Hodgkins and I'm tonight's host. Today's topic is mesothelioma and our presenter is Dr. John Marr, Consultant in Clinical Immunology from the Biomedical Research Centre at St Guy's and St Thomas's NHS Foundation Trust and King's College London. John will be presenting for the first part of our programme, then once the presentation is complete we will go on to answer any questions you might have. If you would like to ask a particular question during the online surgery, please type it into the box on the right-hand side of the screen. Only the panellists will be able to see those questions. John will be answering questions verbally, so we won't be typing any replies. We'll endeavour to answer all your questions, but unfortunately it may not be possible to cover them all in the time we have. If your question goes unanswered, we can still call the helpline on 03000 030 555, and you can speak to one of our dedicated team there. Lines are open from Monday to Friday, 10 a.m. to 6 p.m, and calls are charged at a local rate. That's all from me now, and I'm pleased to hand over to our presenter, Dr. John Marr.
So thank you very much, Vicky. So I'm John Marr. I'm an immunologist, and I'm going to talk a little bit about mesothelioma. So mesothelioma is a cancer which occurs in the natural cavities within the body. So by cavities I'm referring to what's known as the pleura. This is a sac within which the lungs are naturally found. Another such sac is called the pericardium, within which you find the heart. And the peritoneum is the large sac within which the intestines are found. And these sacs are the sites in which mesotheliomas may occur. Now the commonest place in which mesothelioma occurs is in fact in the chest cavity, the so-called pleura. Now the pleura, as I say, is a sac, and it comprises two different layers. Shown in green on the left of this slide, you can see the visceral pleura, which is intimately attached to the lung structure itself. But there is an outer component to the sac called the parietal pleura, and this is more closely opposed to the inside of the chest wall. And it's between these two layers that pleural mesotheliomas grow, as indicated in the cartoon on the right of this slide. You can see that these tumors slowly encase the structure of the lung, and they can also grow into the groove between the lobes of the lung. Now we know quite a bit about what causes mesothelioma, and way out in front is the link between inhalation of asbestos dust or fibers and the onset of this disease. Asbestos has been known for a long time, and the term itself is from an ancient Greek word referring to its unquenchable properties, and in fact that's why asbestos was used for so long as a building material, and I'll talk more about that later. However, the association between asbestos and the onset of this cancer is typically characterized by a long latent period. In other words, there can be an interval of 30 years or more between inhaled exposure to asbestos dust and the onset of this cancer. Now, while asbestos is by far the strongest risk factor for the onset of mesothelioma, there are other factors which may also conspire in the development of this cancer. Genes, as is typical of most human diseases, are important risk factors for the development of mesothelioma. Exposure to radiation may also predispose to the development of this cancer, as indeed may certain viruses, which are relatively poorly understood. But once again, I would emphasize that way out in front is the link between exposure to asbestos, inhaled exposure to asbestos dust in particular, and the onset of this tumor. Now, mesothelioma is a big problem throughout the world, and in the United Kingdom, over 2,500 people were diagnosed with this cancer in 2010, the last year for which statistics exist, and almost a similar number of individuals died of this cancer in the same year. Now, in fact, these numbers are ever-changing, as you can see on this slide, where you see the total number of deaths that occurred in 6 major countries throughout Europe as a result of mesothelioma between 1970 and predicted mortality in 2030. And you can see that mortality is changing over this period, reflecting the prevalence with which asbestos has been used industrially in particular over the course of the 20th century. So another way of putting this is that there is in fact an epidemic of mesothelioma which is occurring and which is predicted to peak sometime between now and the year 2020 throughout Europe. after which it's predicted that that mortality rates will fall because of the increasingly stringent control measures in use throughout many countries in the world with respect to asbestos. Now, if you look at the onset of mesothelioma throughout the United Kingdom, you can see that there are certain hotspots, and these are typically industrialized areas, industrialized cities in which shipbuilding has been a particularly important local industry, and also other areas in which heavy industries in which asbestos use was prevalent. Now, we've known about the link between asbestos exposure and mesothelioma for at least 50 years. This paper here was published by a group in South Africa where asbestos was heavily mined, and in fact was the first clear link between exposure to asbestos and the later development of mesothelioma. So what is asbestos then, and how does it give rise to mesothelioma? So asbestos is a naturally occurring mineral. It's based, it's derived from silica, and it occurs as naturally occurring fibers. And people have known about asbestos for several thousand years. In fact, people have been mining this mineral for over 4,000 years. It used to be referred to as the miracle mineral, in fact, of ancient Greece because of its many positive attributes as a building material. Now, there are a variety of different types of asbestos, and these can be referred to by their natural color. So white asbestos, or chrysotile asbestos, represents 95% of the asbestos that's currently used throughout the world. And as I'll talk about a little later, white asbestos is still mined and used widely throughout many countries of the world, although not in the UK. White asbestos is probably the least risky of the three different main types of asbestos, but it is undoubtedly still dangerous and linked with cancer. Blue asbestos and brown asbestos are the most dangerous forms of asbestos, and these are also known as crucidolite and amosite, respectively. So what you see on this slide is a microscopic image of white asbestos, and you can see that it forms these snake-like or serpentine fibers, whereas by contrast blue and brown asbestos forms these needle-like structures, and these structures in particular, when they are inhaled into the lungs, are extremely difficult for the body to get rid of. So how big is the problem of asbestos-related disease? Well, in fact, asbestos exposure is the single greatest cause of work-related death in the United Kingdom, and asbestos exposure accounts for more deaths each year than occur as a result of road traffic accidents. It's estimated that between the 1940s and 1980 that approximately 40% of the workforce were naturally exposed to asbestos in the course of their work. And that is generally much more dangerous than the exposure to asbestos that may occur in the home, because of course in the workplace asbestos may be manipulated, giving rise to release of fibers. And at the present time, asbestos is still present in many buildings, and I'll talk a little bit about that later as well. So why have people been using asbestos for all this time? Well, as I've intimated earlier, asbestos has many properties which make it a very useful material in industry. It's resistant to heat, it's resistant to chemical and electrical stress, it's got very high tensile strength, meaning that if you stretch it, it's very, very robust. But on the other hand, it's quite flexible. So if you're using it to insulate a material, for example, it's easy to wrap it around that, to wrap it around pipes or boilers, for example. So as a consequence, there have been several industrial and domestic applications of this mineral. And indeed, it's been estimated that over 3,000 products contained asbestos at one time. Asbestos has multiple uses in the construction industry, and is also was also widely used in insulation, particularly in train engines and in shipbuilding. The automotive industry used asbestos particularly in clutch and brake linings, and most electricity power stations were also full of asbestos. This slide here just illustrates some of the places that asbestos can be found in the family home. And you can see in order to insulate flooring or walls, or to insulate boilers or pipes, or indeed ceilings, that asbestos was very commonly used in buildings, particularly between the 1950s and up to as recently as the year 2000. However, many people can become quite alarmed when they realize this. However, most asbestos-containing materials found in the home are generally not considered to pose a health risk because, as I mentioned earlier, the key risk is when asbestos is released into the air in the form of fibers that can be inhaled. So generally the best thing, if asbestos is present in the home, is that it should not in fact be touched, because any manipulation of that may cause the release of fibers, which in turn could be dangerous. And both the Health and Safety Executive and local councils provide a lot of guidance on how asbestos should be dealt with in the home. And indeed this website, sponsored by BLF, will also provide further information on that particular topic. Another example where large quantities of asbestos fibers were released into the atmosphere recently was the terrorist attacks that occurred on 9/11. And it's been estimated that when the Twin Towers collapsed, that between 400 and 2,000 tons of asbestos fibers were released into the air, causing direct exposure to 500,000 New Yorkers. And indeed many of the rescue workers who were first on the scene at Ground Zero have suffered from a variety of respiratory illnesses, including some cases of mesothelioma since 9/11. It's estimated that pretty much all occupants of New York City, particularly in the downtown Manhattan area, were exposed to asbestos as a result of the terrorist attacks on 9/11. Now, asbestos use is tightly regulated in the UK, and indeed it was Denmark who took the lead in regulating the use of this mineral back in 1972. And as the 80s and the 90s and the noughties progressed, increasing numbers of countries have introduced a complete ban on the use of all forms of asbestos. However, unfortunately, that situation does not pertain throughout the world, and asbestos continues to be mined in a number of countries. In fact, in Canada, asbestos mining took place until as recently as 2010. This slide here shows you a large asbestos mine in a town which is in fact called Asbestos. And I'm glad to say that with a recent change of government in Quebec, within which the town of Asbestos is found, that this mine is no longer operative. However, asbestos continues to be mined in several other countries. This is the Asbest mine in Russia, and as you can see, it's an absolutely enormous mine. And indeed, asbestos mining is conducted very widely throughout Russia, where almost 1 million tons of asbestos are mined each year. And this has great significance to the economy in Russia because it employs approaching 500,000 people and generates over $800 million in revenue per annum. So the asbestos industry has significant support from the government in Russia, and when I talk about asbestos mining in this context, I'm talking in particular about the mining of white asbestos or chrysotile. In order to support this activity, a live chrysotile organization, in fact, has been set up in order to provide reassurance to individuals about the safety of this approach. This is from their website, and indeed if you click on the Health and Safety button on that website, you will read a great deal of information arguing that exposure to white asbestos is safe. However, this information would not be consistent with the general medical view in the Western world, which is that white asbestos or chrysotile certainly causes lung cancer and probably also causes mesothelioma as well, although exposure to white asbestos is considered to be less dangerous than either blue or brown asbestos. Asbestos was also widely mined in other countries. This image here is taken from the town of Wittenoom in Australia, where blue asbestos was mined. And you can see two children in this image who are playing in a sandpit of blue asbestos. Both of these children, in fact, died of mesothelioma before they reached the age of 40. And the town of Wittenoom is now essentially a no-go area in Australia. Indeed, if you look at the road signs in the area, you can see that the place name has been removed from them. And it is a highly dangerous area to visit. So at the present time, 52 countries throughout the world have banned the use of asbestos, although several countries continue to both mine and use this mineral. China is the biggest consumer of asbestos, and use is also increasing quite dramatically in India. And these are images that were provided to me by my colleague Astero Klabatsa when she was in India, showing the local inhabitants openly working with and indeed cutting asbestos fibers, asbestos material, releasing these fibers into the atmosphere. However, the news is not all doom and gloom, because this slide shows a protest in a town in Bihar in India where there was a move to introduce an asbestos cement factory. And indeed this would have been of great benefit to the local economy, but it was the teenagers who had learnt about the hazards of asbestos in school who in fact mobilized public opinion against the opening of this cement factory, and that factory has not opened since that time. So much for asbestos. What about the illnesses that this mineral can cause in man? And in fact, asbestos can cause several illnesses in addition to mesothelioma, from benign conditions such as collection of fluid in the pleural sac, or the formation of thickening, so-called plaques, of the pleura. Such thickening may be found diffusely throughout the pleura as well. Inhalation of asbestos fibers into the lungs can also cause scarring in the lungs, a disease called asbestosis, which can also be life-threatening. Asbestos, in addition to causing mesothelioma cancers, can also cause a commoner form of lung cancer, and indeed there is some evidence that it may also be a factor in occasional cases of ovarian cancer. So how does asbestos actually give rise to mesothelioma then? Well, what you see in this image is a single asbestos fiber, probably a blue or a brown fiber, the needle-like fibers that I showed in an earlier image, which is trapped in the lungs. And you can see that the fiber is surrounded by small blue blobs, and these blue blobs, in fact, represent a type of cell of the immune system called a macrophage. And macrophages are basically professional scavenger cells, so their role in life essentially is to ingest material that has been, in this case, inhaled into the lungs in order to eliminate it. So for instance, most people who live in cities will inhale a lot of pollution over the course of their lives, and the job of the macrophages in the lungs is to gobble up this material, as it were, in order to dispose of it. However, an asbestos fiber, as you can see in this image, is much too big for these cells to ingest. You can see that there are probably 20 macrophages stuck to the outside of this fiber, and they simply don't have any prospect of eliminating the fiber. It's essentially insoluble. And when this situation happens, these macrophages essentially become frustrated. They're constantly trying to ingest this material and failing, and that triggers a cascade of inflammation in the lung, which ultimately is damaging. And this process keeps going on and on and on over many years, and indeed when mesotheliomas occur, about 30% of the tumor mass is in fact these macrophages, trying in vain to eliminate the asbestos fibers. When asbestos gets into one of these macrophage white blood cells, it activates this spider-like creature here, which is known as an inflammasome. And inflammasomes are naturally produced by these white blood cells in order to trigger inflammation. And the way that they do that is the inflammasome causes the activation of molecules which are called interleukins. And these interleukins, the function of these proteins in life is simply to trigger inflammation. Asbestos is a very powerful natural activator of the inflammasome. So it signals to the macrophage that inflammation needs to be propagated. Normally when inflammatory responses occur over a short period of time, they're beneficial. So inflammatory responses can help to get rid of bacteria from the lungs, for instance. But if you have a long-term grumbling inflammatory process going on over years, as is the case with asbestos exposure, that is harmful because inflammation over a long period of time can actually cause several detrimental changes, including damage to the DNA within the mesothelial cells. In other words, the cells that line the sacs, the pleura, the pericardium, the peritoneum, etc. And this DNA damage, of course, can be dangerous and can set the seeds for the development of cancer. Now, the changes that occur in the mesothelial cells as a result of this chronic inflammatory process, well, there are several such changes. What you see on this slide here is the surface of a mesothelial cell, and you can see, I think, 8 different so-called receptor molecules which are found on the surface of these cells. Now, these receptors basically serve the role of enabling the mesothelial cells to see what's going on in the outside world. But as a result of the chronic inflammatory process that goes on in the lungs, and in particular in the pleural sac, these receptors become increased. They become upregulated on the surface of the mesothelial cells. And as a result of that upregulation, these receptors can deliver powerful growth-inducing signals to the mesothelial cells, telling them to replicate themselves, to make more copies of themselves. In the first instance, this kind of change may give rise to the pleural thickening, pleural plaques that I mentioned earlier on, but after many years, this can set the seed for the development of cancer, in other words, mesothelioma. Several other genetic damage, genetic lesions really, have been described in mesothelioma, and I've summarized some of these on the bottom of the slide. Basically, these genes naturally serve as brakes, stopping cells from dividing. And of course, if you disable the brakes within the cells, it provides a stimulus to these cells to replicate themselves, to make more copies of themselves. And this, of course, is a dangerous situation because it can predispose to cancerous change. So the net effect of this chronic inflammatory process triggered by asbestos fibers in the pleural sac is to cause DNA damage within the mesothelial cells, and ultimately mesothelioma may occur. Mesothelioma is a very difficult disease for clinicians to treat, and there are several problems and challenges that this cancer poses. The first problem is that patients with mesothelioma may have little or no symptoms for a long period of time, so consequently the disease may be quite advanced when it comes to medical attention. The second problem, as I'll allude to later, is that the best form of surgery for this cancer is a subject for some controversy. Chemotherapy, using 2 or more chemotherapy drugs, can be beneficial in some patients, but generally the results are quite modest and of relatively short duration. And the other traditional therapy that people tend to use in cancer, i.e. radiotherapy, is also challenging because it can be difficult to deliver adequate doses to the tumor owing to the fact that you've got key organs such as the heart, the lungs, the spinal cord close by, limiting the dose that you can deliver. These are some of the symptoms that patients with mesothelioma of the pleura can present with. But as you can see, people with many other respiratory diseases can have some of these symptoms as well. So it can be difficult for the penny to drop in terms of the fact that there may be a disorder such as mesothelioma going on. In order to diagnose mesothelioma, I guess the first type of investigation that will often be carried out is some kind of imaging. So chest X-ray or a CT scan of the chest may be carried out in the early stages. Other imaging techniques such as magnetic resonance imaging or MRI may also be carried out, particularly if there is a question mark about definitive surgery being carried out in patients. Sometimes ultrasound can be useful in these patients, particularly to identify the right site from which a biopsy may be taken. And in some cases, scanning with a relatively new scanning technique called PET can also be useful. And I'll show you some examples of how these imaging techniques can aid in the diagnosis of mesothelioma in the next 5 or 6 slides. So this is a typical chest X-ray from a patient with mesothelioma, and you can see that low down on the right-hand side of the image, in other words in the left lung here, you can see that there is a white area which has a smooth upper limit rising up into the armpit. And this is what we call a pleural effusion, or in other words a collection of fluid in the pleural sac. And in some cases this can be a manifestation of a mesothelioma. This is an example of a CT scan taken from a patient with mesothelioma, and in this image you can see the tumor here low down in the left lung, which you can see on the right hand side of the image, and the tumor is actually encasing the lung, and in fact is also present on the inner aspect of the lung. You can see some of the lymph glands in the middle of the chest are also enlarged because the tumor has spread to affect these lymph glands. Here's another example of a CT scan where the tumor is now on the other side of the chest. You can see that the tumor has encased a darker area in the middle, and this is a collection of fluid, a pleural effusion, on this occasion on the right-hand side of the chest. If surgeons are contemplating carrying out an extensive surgical procedure in an attempt to remove all of the tumor, they may sometimes also carry out this type of scan called an MRI scan, magnetic resonance imaging. And as you can see in this panel here, panel C, MRI scanning can give you very good information about the extent of spread of the tumor, enabling the surgeon to have better information before they bring the patient to theatre in an attempt to debulk the tumor. You can see, for example, in panel D here, the considerable information that an MRI scan can give about the extent of the tumor, which, as you can see on this occasion, is encasing the lung within it. More recently, PET imaging has also been carried out, and the idea behind a PET scan is that you administer a radioactive form of sugar to the patient through the bloodstream. And the idea behind this is that the cancer cells are more avid for the sugar than are the healthy cells. They tend to take up the radioactive sugar to a greater extent, and as a consequence you can see that the tumor literally glows on this scan. And this can be very useful to monitor the effectiveness of a treatment that you administer to a patient, comparing the PET-CT image before and after the therapy is administered. Now, imaging is useful in aiding the diagnosis of this condition, but at the end of the day, to make a definitive diagnosis, you need to obtain tissue from the tumor. And this can be carried out in a number of ways. A VATS procedure, or video-assisted thoracoscopy, is a procedure in which a surgeon places an endoscope into the chest cavity so that they can actually visualize the inside of the pleural space. And when they visualize that space, that allows them to identify areas which look clearly abnormal, from which biopsies can be taken. This procedure is also useful because the surgeon can introduce a chemical into the chest space in an attempt to prevent the reaccumulation of pleural fluid, because of course pleural fluid can cause a lot of symptoms in these patients, such as breathlessness in particular. A second way in which tissue can be obtained is to obtain a pleural biopsy guided by a CT scan or an ultrasound scan. And an alternative way in which the diagnosis may be made is to take a sample of pleural fluid, although this may be a little bit more difficult than taking a biopsy in terms of being absolutely definitive about the diagnosis. So this image here shows you an example of the VATS procedure that I mentioned earlier, where the surgeon has placed an endoscope into the chest cavity and can see clearly abnormal whitened areas which represent mesothelioma. And so because the surgeon can see the extent of the disease, this allows him or her to take biopsies from the relevant areas. When biopsies are taken, they will be sent to the pathology lab, and this allows the pathologist to identify three major types of mesothelioma, so-called epithelioid, sarcomatoid, and mixed. And you can see typical examples of these three types of mesothelioma in this slide. However, it's not by any means as simple as highlighted here, because sometimes different types of cancers may assume a similar type of appearance to the typical mesothelioma samples that I've shown on this slide. So in order to add further weight to the diagnosis, typically pathologists will add antibodies to the tissue sections which identify characteristic protein molecules which are found in mesothelioma but not in other cancer types. So often, to make an absolutely definitive diagnosis of mesothelioma, several steps need to be undertaken, from imaging through taking biopsies, through performing these special stains with various antibodies in order to be absolutely cast iron about the diagnosis. This is an example of some pleural fluid from which a sample was taken, and you can see in the middle of the slide a collection of perhaps 40 or 50 cells, some of which are very abnormal. I mean, you can see a cell over on the side here which has got 3 so-called nuclei. This is a completely abnormal cell. This highly clustered appearance of cells is often seen in patients with mesothelioma. But as I mentioned earlier, in order to really consolidate the diagnosis, there may be a necessity to take a biopsy as well and perform these additional pathological tests. So how is mesothelioma treated in the 21st century then? Well, generally the approach that will be taken is a so-called multimodality approach. In other words, a combination of approaches may be used in an individual patient. Surgery is commonly carried out. I've already mentioned the VATS procedure, whereby a surgeon will place an endoscope into the chest cavity to make a diagnosis, and this will also allow the surgeon to introduce a chemical in order to prevent the recurrence of fluid, of pleural effusion, which affords symptom relief to the patient. That procedure is known as a pleurodesis, and in about 3/4 of cases will give relief of symptoms to patients. However, in some cases, more extensive surgery will be carried out, particularly in younger, fitter patients, with the goal of debulking the tumour. In the most extensive case, a so-called extrapleural pneumonectomy will be carried out, whereby the surgeon attempts to remove all of the visible disease together with the pleural sac, the pericardial sac, that's the sac that surrounds the heart. The diaphragm is removed on that side of the chest, and the lung is removed as well. So this is a very extensive surgical procedure. But recent studies have suggested that the quality of life of patients who undergo this procedure may not be significantly better than with a lesser surgical procedure. So an alternative approach is a so-called pleurectomy and decortication, where the surgeon removes as much of the disease as possible and the pleura, but leaves the lung in situ within the chest. Chemotherapy is also commonly used to treat patients with mesothelioma. A number of chemotherapy drugs can be useful in this disease, particularly platinum-based drugs such as cisplatin or carboplatin, and antifolate drugs, most notably pemetrexed. But a number of other chemotherapy drugs can have activity in this disease, such as gemcitabine or vinorelbine. Whilst a significant proportion of patients will benefit from chemotherapy, the benefit afforded tends to be relatively short-lived and may be measured in months, really, rather than years. The third approach in the multimodality management of mesothelioma may involve the use of radiotherapy. And this can be useful also to improve local control of the disease within the chest cavity or to relieve symptoms, to palliate symptoms. More recently, a form of radiotherapy has emerged known as IMRT, and this enables the radiotherapist to deliver higher doses of radiation in a precisely targeted manner to the tumor. I mentioned earlier that one of the challenges with radiotherapy is the risk of side effects because of radiation of the lung or the heart or the spinal cord, but with IMRT, more precise delivery of radiation may be possible in some cases, so this may assume an increasing role in years to come. However, it's clear that with current management that there is still a very large unmet need for better treatments to achieve better control of this disease. And several experimental approaches are under development for this cancer. A number of these involve the delivery of experimental agents into the chest cavity, whether it be chemotherapy or gene therapy. An alternative approach that is in development are so-called targeted therapies. In an earlier slide I showed that perhaps 8 or 9 different receptors may be expressed at higher levels in these cancer cells, so consequently investigators are testing the effects of blocking these receptors in an effort to improve patient outcome. So far, however, this kind of approach has not yielded very promising results. Yet another area that we are particularly interested in involves the use of immune therapies, trying to boost the patient's immune system so that it will then attack the mesothelioma cancer. And a whole slew of different immune therapies are under development for this cancer, including vaccines or the use of inflammatory proteins called cytokines, or indeed the use of cells of the immune system. And it is the use of cells of the immune system that we in our lab are trying to develop as an experimental therapy for mesothelioma. Naturally, we all have white blood cells circulating in our bloodstream called T cells, whose role it is to attack and treat infection. And the way in which these T cells do this is because they have molecules called receptors which allow them to see infected cells and to kill those cells as a result. So what we are trying to do is to engineer artificial receptors called CARs, or chimeric antigen receptors, and an example of a CAR is shown in the middle of this cartoon. And the idea of the CAR is that if you can introduce these molecules into T cells, you can equip them with the ability to see and attack cancer cells, such as mesothelioma. In this slide here, I have 2 movies, in fact, which show what happens when we develop a CAR in the laboratory and take those T cells expressing the CAR and mix them with cancer cells. So the movie that is running at the moment shows you what happens when you have a CAR that has an intact outer part, so it can bind onto a cancer cell, but it does not have an intact inner part, and consequently the T cell does not receive any information telling it to attack the tumor. By contrast, if you have the fully intact CAR, you can see that the T cells now destroy the tumor monolayer. It essentially melts away, and you see the formation of these large clusters within the middle of the culture, representing activated T cells mixed in with dead and dying tumor cells. Now, of course, it's one thing to do these experiments in a tissue culture dish. It's quite another thing to get this kind of technology to work in a patient with an advanced cancer, such as mesothelioma. This slide here shows some experiments that we have carried out with 3 different mesothelioma cell lines called H28, MM98, and REN. And you can see the appearance of the cancer cells stained up with the blue dye on the immediate left of the slide. If you take T cells and mix them with these cancer cells, the T cells will ignore the cancer cells, so you can still see the blue staining present in the wells. If you introduce a CAR which cannot recognize the mesothelioma cells, similarly you still see the intact tumor monolayer, whereas if you introduce a CAR into these T cells that can in fact see the mesothelioma tumor, you can see that the tumor monolayer is completely destroyed within these cultures. Now, the small amount of research that I've told you about so far is carried out in a research environment where we grow the cells in plastic dishes, such as are shown in this part of the slide. But in order to make CAR-engineered T cells to treat patients with mesothelioma, we instead need to move to a different environment, a so-called GMP facility, within which we can grow T cells derived from the bloodstream of patients in order to develop this type of experimental therapy for clinical use. And when we do this type of thing, we end up with bags of these T cells, such as you can see in this slide here, so-called T bags. And when you look at these T cells in the bag down the microscope, you see this appearance, which is shown on the right-hand side of this slide. So the research that we carry out is funded by the British Lung Foundation, so we would all like to extend a big thank you to the BLF for this support. And I'd also like to thank all of the people listed on this slide who work both in the laboratory and in the clinic in our CAR-based research activities. So thank you all very much once again.
Thank you, John. That was really, really fascinating and very comprehensive. If anybody does have any questions, then in the box that's on the right hand of your screen, if you could just type them in now, and we'll be able to ask John live as he's here and he's joined us very kindly. I do have a couple of questions already. There's one from Jenny, and she asks, how do you think the new treatment options that you spoke about might be developed for patient benefit?
Okay, so I just touched on that very briefly at the end of the talk, but in fact we have been working on the CAR approach for a number of cancers for the best part of 10 years or so. We have developed several of these molecules over that period of time, and one of these molecules has reached the point that we are only months away from starting a clinical trial in patients. Now that CAR has been developed for patients with a different cancer, head and neck cancer, but in fact the information that I think we'll obtain from that clinical trial could have significant implications for mesothelioma, because it may well be that we could use a similar approach down the line in patients with mesothelioma, putting these engineered T cells into the chest cavity of the patient. So the other reason that that trial could impact upon mesothelioma is that it allows us to develop the kind of manufacturing processes that we need in order to make these cell products at a clinical grade for patients.
Okay. We also have another question from Michael, who asks, are there any other new treatments on the horizon in this area?
So, I mean, absolutely. There's a great deal of research going on to develop new types of treatments for mesothelioma, in particular because the existing therapies have only really a modest effect. So in addition to the kind of immune therapies that I've talked about, there's also a lot of interest in developing so-called gene therapies for mesothelioma. For example, several groups are exploring the use of viruses which can be genetically engineered so that these viruses can in fact infect the cancer cells and cause their destruction. So I think gene therapy is also likely to be a promising area in years to come. And one of the attractions of mesothelioma for both immune therapies and gene therapies is the fact that there is a real prospect that you can introduce the therapeutic agent directly into the chest cavity, into the pleural space, so that you can actually deliver the therapeutic agent directly to the site of the tumor.
Great, thank you. Sorry, for those of you who didn't hear, that was Michael's question, and he asked, are there any other new treatments on the horizon in this area? Thank you very much, John. I'm afraid that's all we've got time for this evening. I hope everyone's found today's online surgery useful useful. Just like to say thank you again to Dr. John Marr, who's a consultant in clinical immunology, well that's a hard one to say, for the Biomedical Research Centre at Guy's and St Thomas's NHS Foundation Trust and King's College London. We won't be able to answer any more questions now, so I apologise if any have gone unanswered. You can still call our helpline on 03000 030 555. That's open Monday to Friday, 10 a.m. to 6 p.m. The helpline team can provide advice on all sorts of issues related to lung disease and lung health. And as John mentioned in the presentation, there is our Take 5 and Stay Alive campaign website, which is take5stayalive.com, where you can go and have a look if you're planning on doing any DIY in your home. And if you know anybody that is doing any DIY, do direct them there. There's also information on mesothelioma on our website, www.blf.org.uk. A recording of